A recipe guaranteed to amaze and delight. To 1 jar of pond water, add some decomposing leaves, and let age for several days. After day 3, a characteristic odor indicates that bacteria are multiplying. On day 4, a population of protists appears just beneath the surface. By day 5, there are plenty of organisms for observation. Add a fragment of the decomposing plant material and observe. What is it about decaying material that is so attractive? Shake them up. In a minute, they are back. How can these observations be explained? Observing Paramecium under higher magnification shows what it eats and how the food is digested. Along one side, the cell has a channel, the oral groove, ending in a cytostome, the cell mouth. Here, bacteria are funneled into a short gullet where they are engulfed. Within the vacuole, the bacteria are broken down into their building block molecules, and these are released for use by the cell. An acidic environment speeds up enzymatic digestion. In humans, glands in the stomach lining produce hydrochloric acid, making enzymatic digestion more efficient as food progresses down the intestine. Smear a small amount of neutral red stain on a slide and allow the alcohol solvent to dry. Add Paramecia. As the stain is taken up, regions that are acidic turn red, the food vacuoles. The small red staining particles are lysosomes, acidified canisters that transport digestive enzymes from their manufacturing sites to the food vacuoles. Containment is vital. If these powerful chemicals were to escape, they would digest the Paramecium from within. In Paramecium, most of the food is broken down and absorbed by the cell, but non-digestible solids accumulate. These solid wastes are eliminated through a temporary opening in the cell membrane. Chemical waste products are stored in the form of crystals that show up in polarized light. Water entering the cell by osmosis is collected by a network of tubules that connect with a contractile vacuole. When full, the central reservoir empties through a pore. Most Paramecium species have a contractile vacuole at each end. Paramecium's outer covering is amazingly flexible. Cilia project through this flexible skin, setting up waves of motion. Cilia propel the cell, or help anchor it where bacteria are plentiful. Once on a feeding station, cilia can set up powerful feeding currents. The whipping action comes from molecular motors energized by ATP. A cilium is made up of microtubules. The outer ones have little protein claws that, when energized, pull on an adjacent microtubule. This molecular clawing is the basis for the power stroke recovery stroke seen in the ciliary beat. Paramecium's day-to-day activities are controlled by its macronucleus, the large bluish structure. When the cell divides, its macronucleus pulls in two, distributing multiple copies of Paramecium's chromosomes to each of the offspring. After many reproductions, the mix of chromosomes can become unbalanced, and the Paramecia become sickly. Generating a new balanced set of chromosomes is accomplished through a sexual process called conjugation. When paramecia belonging to different mating strains come together, there is a sort of courtship in which the paramecia feel each other with their cilia. If compatible, they fuse together and exchange micronuclei. This tiny structure contains a pristine set of chromosomes from which a new macronucleus can be generated. The mating partners now contain genetic information from both parent strains, improving the chances for genetic variation among the offspring. This paramecium is a farmer, an algae farmer. The relationship is symbiotic. The alga provides products made by photosynthesis, while the host cell supplies its guests with raw materials and a place to live. However, if kept in the dark, Paramecium bursaria breaks off the partnership and digests the algae, and then goes hunting for other snacks. Paramecium is eaten by all sorts of small organisms, but one is particularly relentless, Didinium. As prey are depleted, an increasingly hungry Didinium will lay down a protective covering, forming a cyst. It will remain dormant until the prey population rebuilds. A stinking brew culture encourages large numbers, but relatively few kinds of organisms. To see a greater diversity of ciliates, hunt for them in places like roadside puddles. One of the protists living on duckweed rootlets was first observed 300 years ago by Leeuwenhoek, a stalked ciliate that takes its name from the tiny whirlpools it creates, Vorticella. Vorticella reproduces by fission, but which daughter will retain the stalk? The first hint comes when one cell develops a band of swimming cilia. This daughter will break free and reattach, not just anywhere, but near its sibling. This homing behavior ultimately produces clusters of vorticellids. A furry coating of vorticellids can create habitat for intruders. One way to deal with unwanted guests? Relocate. At times, all of the individuals in a Vorticella clump will change from bell-shaped cells to these elongated migrants.